Good afternoon or good morning, everyone. Welcome to the AIR Community Seminar Series. I'm Incarnita Mariotti Ferrandis. I'm the chair-elect of the AIR Community, and I'm going to chair this seminar today. I'm really happy to welcome our two speakers, Aaron Michels from the Barbara Davis Diabetes Center from University of Colorado. So Aaron, as you saw, will discuss about TCR repertoire in type 1 diabetes and health with a focus on young repertoire, so young people repertoire. And our second speaker is Julien Limenitakis from the University of Bern in Switzerland. And his talk is going to be about microbiota, diet, and the effect on B-cell repertoire. So we will have first Aaron for a 45 minutes discussion, presentation, and followed by 15 minutes discussion. So please turn off your microphone and cameras. And during the presentation, the question session, you can raise your hand to ask your question. So we expect the question to happen only during the discussion session. So without more spending time, Aaron, the stage is yours.
Thank you, Incarnita, for that kind introduction and really the opportunity to speak to the AIR community. I'm really excited to share some of the work that we've done, obviously sequencing T-cell repertoires in young, healthy children and those that progress to type 1 diabetes. So our center is a large center in the United States that sees children and adults with type 1 diabetes. And I'm a physician and scientist there. So I do see patients a little bit, but focus a lot on research. So I thought I'd do a little bit of background on type 1 diabetes before getting into all of the T-cell receptor sequencing. So type 1 diabetes, it's also referred to as childhood onset diabetes, juvenile onset diabetes. And really, at a high level, what we know about the disease in 2023 is it is a common and increasing autoimmune disease. There's heterogeneity in who develops the disease. Obviously, children will develop disease, but a lot of adults, and especially young adults, are newly diagnosed with type 1 diabetes. The disease is predictable. We can measure type 1 diabetes associated antibodies or pancreatic islet autoantibodies and know who will go on to develop clinical type 1 diabetes needing treatment with insulin and blood sugar monitoring. In the last year, we now have our first immune therapy to treat diabetes to delay the onset of clinical disease. This is teplizumab or TZIELD, which is an exciting time and really a change in paradigm for how we think and treat type 1 diabetes clinically. So type 1 diabetes, when I was in medical school and learning, it was almost always, North America, Europe, and Australia, but that really isn't the case anymore. So this is incidence in children, and obviously darker the color means more cases. But as you can see, there really is a global incidence and predominance of type 1 diabetes. And what's been really shocking is the incidence just continues to rise at about 3 to 5% every year and has doubled in the last 20 years. And shown here are just some countries and the state of Colorado where we've tracked the diagnoses of diabetes very well. And you can see these lines just continue to increase over time. And with the very, very high incidence, large incidence occurring in the last 20 years, probably very likely related to environmental factors. There is a strong genetic basis for type 1 diabetes, but really hard to put enough genes in the environment over a 20-year period to see, increasing trends like this. So we know a lot about the natural history of type 1 diabetes. It has always been termed a chronic autoimmune disease. And this figure, first presented by George Eisenbarth, Keeps going. It really is a roadmap. So each individual starts out with a given beta cell mass. Islets in the pancreas, which encompass about 1 to 2% of the pancreas. There is genetic risk, genetic predisposition. It is a polygenic disorder. Something or some things in the environment are very likely needed to trigger autoimmunity, which we are best able to measure at the current time with antibodies. And we'll talk a little bit more about those. Time is ambiguous, it goes on. And generally, we start to see patients here where they present with clinical symptoms, increased thirst, needing to urinate, weight loss, and this marks the need for insulin treatment. As I mentioned earlier, we now have immune therapies, antibody targeting anti-CD3 on T cells that can, when used earlier in the disease course, delay the onset approximately 3 years before needing treatment with insulin. So what do we know about the self-antigens in type 1 diabetes? Quite a bit now. So this is an islet in the pancreas that is actually stained with serum that fluoresces, having islet antigens. We know these are now secreted, things like insulin, glutamic acid decarboxylase, islet antigen 2, and zinc transporter 8. All of these three are intracellular proteins really needed for packaging and secretion of insulin. And there are likely other targets that we don't know for antibodies. But with 4 antibodies, we really can track and predict which children will go on to develop diabetes. So this is just an individual case, and this comes from a prospective birth cohort study that we did in Colorado. And each symbol is a time where we measured 3 antibodies at the time here to GAD, islet antigen 2, and insulin. So you can see at 3 to 6 month intervals until about the age of 8, when all 3 of these antibodies became positive. There's then a nice delay or lag time where the antibodies do remain positive, and then clinical diabetes develops roughly in this individual 6 years later. So with that knowledge and the ability to measure for antibodies in the disease course, again, birth cohort studies of those at risk in Colorado, Finland, and Germany pulled all the children that had 2 antibodies or more and then followed for progression to type 1 diabetes. And so nearly 100% will develop type 1 diabetes over time. And it's remarkable that this curve and slope, it's roughly 11% every year, go on to develop clinical type 1 diabetes. The curves for each country or state are shown here and are very similar. So antibodies are great predictive markers. The interesting part in type 1 diabetes is the antibodies aren't involved in the disease pathogenesis. They don't coat antibodies, they don't activate complement. They really mark a disease process that is going on. And so this would be the gap. Antibodies predict who will go on at a population level. But at an individual level, I can't tell you an 8-year-old with 2 antibodies will develop diabetes in a year, 5 years, 10 years, or even 20 years. And so we really need a more dynamic biomarker of disease activity. And that's where we have turned to T cells and especially T cell receptors. And so a number of years ago, we really wanted to start sequencing T cell receptors through the stages of diabetes. And from that initial model, the stages have been a little bit more defined now. So stage 1 diabetes really marks 2 or more antibodies, no blood sugar abnormalities. Stage 2, you still have antibodies. You have dysglycemia, we'll call it mild hyperglycemia, not enough to lead to a diagnosis of diabetes. And then stage 3 diabetes is new onset. So you walk in the clinic, high blood sugars, needing treatment with insulin. So our really ultimate goal here was to use disease-relevant T cell receptors as a non-cellular biomarker during type 1 diabetes. And a little more background, we know very well that T cells infiltrate the islets, especially CD8 T cells with the machinery are there and can kill insulin-producing beta cells. So my lab thinks a lot about T cells, the trimolecular complex. So HLA presenting peptide to a T cell receptor. And we've studied this for the last 15 years in terms of HLA molecules. Why do they predispose risk? Why do they protect? What sort of peptides, whether they're native or post-translationally modified, stimulate T cells, and what are those responses? But just shown here, we have a peptide MHC complex. So MHC is kind of in white and magenta. The peptide is yellow, and the kind of shaded area here is the T cell receptor. And so we think about this in a lot of detail, but the T cell receptor sees and contacts different portions of the peptide and also makes contact with an HLA molecule. And so I assume this audience from AIR knows quite a bit about antigen receptors on T cells and B cells, but just suffice it to say there is a large amount of rearrangement to make a very diverse repertoire of T cells. We have billions of T cells to respond to millions of antigens, and we have much more limited number of HLA molecules in a given individual. And really for this talk, we're focused here on DNA, especially of the T cell receptor beta chain, and sequencing those. So to start our work in I will say children with health and diabetes, in Colorado, we have the DAISY study, the diabetes autoimmunity study in the young. And in this study, which started back in the 90s and has followed children over time, we set out to identify children at risk for diabetes. So either those that had genetic risk, so think HLA genes DR3 and or DR4, DQ beta 1 marks DQ8, or just had a first degree relative, whether a parent or a sibling with type 1 diabetes. So we screened over 30,000 individuals, identifying those at high risk, and followed those at 3 to 6 month intervals to the best of our ability over time. And we now have approximately 106 that a little over 100 that have gone on to develop clinical type 1 diabetes. So we decided to take a subset of these DAISY individuals at 4 time points. And we actually had cryopreserved Buffy coats from a lot of these subjects where we could isolate DNA. We used adaptive sequencing, so DNA sequencing of the T cell receptor beta chain. And we tried a nested case control cohort design. So we have 29 cases here, sampled at 4 times over life. Time point 1, we tried early in life, so age down here at the bottom. A lot of these circles are, right around 1 and under 2 years of age. Time points 2 and 3 for the cases, we tried to go right before and right after the development of those islet autoantibodies. And then time point 4 was right before diagnosis of clinical type 1 diabetes. Controls were age, sex, and HLA matched. And we had 25 controls here. So quite a few samples longitudinally over time from a really young cohort, predominantly children and adolescents. And this is just some of their baseline characteristics at the 4 time points. Controls and cases, mean 1.9 years of age, 2.1, 5, 6, and then right around 11 and 12 for time point 4. Gender, there is a male predominance for type 1 diabetes. Race, it is predominantly non-Hispanic whites that can develop type 1 diabetes. And then our cases definitely developed antibodies. None of our controls did, and were monitored over time. Since we're talking about T cells and T cell receptors, we did want to try to the best of our abilities to match HLA, again, outbred human population, but here are all the controls and cases with class 2 and class 1 typing. And especially for the high genetic risk genes, DR3, DR4, DQ2, DQ8, there was very good matching between cases and controls, and also pretty good too for A, B, and C in class 1. So we had Adaptive do their immunosequencing, and this is just, again, showing what we're able to find. So these are productive TCR beta chain templates. Controls are blue, cases are red, and these are just numbers by time point. And as you can see, it's nice that the sequencing is remarkably consistent over time. And even in young children, roughly around the age of 2 here, we're able to detect and measure a very good repertoire. So when we started analyzing this data, we obviously wanted to try to find insights into diabetes, but also knew that, boy, T-cell receptor repertoire just hadn't been sequenced longitudinally over time in young children. And so we just decided to look at, I'll say, fairly simple metrics of Simpson productive clonality. And as expected, that increased. So the repertoire is very diverse, low clonality early in life, and that increases over time. In all samples, obviously, it did that in controls and in cases that went on to develop type 1 diabetes as well. And then shown on the bottom are, again, just individual level data. Lighter color is time point 1, darker is time point 4. And so the repertoire, very diverse early in life, becomes more clonal over time, likely from infections and vaccinations that occur. So then we decided to dive into all the V gene, V beta allele usage, and we were really hoping to find some striking differences between cases and controls. And so shown here now are all of our cases at time point 1, 2, 3, 4, and controls. I guess the nice part is we were able to find all these V beta genes and alleles from the sequencing. We didn't have big gaps. But unfortunately, as we looked at this and went, allele by allele, there was really remarkable consistency across cases and controls. And there just really wasn't that smoking gun that said, oh my gosh, it's V beta 3, 5, 14. That is dramatically different between age, sex, HLA-matched controls that never develop islet antibodies or type 1 diabetes versus cases. We still tried to dive into the data from the broad repertoire and just do principal component analysis with different T-cell receptor V genes, saying, can we, early in life, from DNA, prior to the development of antibodies, markers that you're going to go on to develop diabetes, is there any way we can try to identify cases, or in this case, a subset of cases that may develop antibodies and type 1 diabetes later in life? And so there are 11 kind of light red subset of cases here. They're in the same circle at time point 1, 2, 3, and 4. The principal components shown here again are T-cell receptor V genes. And we can see that in this subset of cases that we could identify very early in life, a little bit more TRBV 0403 usage, maybe a little bit less, 28. And again, we can look at how these progress over time. The trends were consistent of, say, more TRBV4, less TRBV 28, a little bit more 30-01. And that was nice to see. But we, again, wanted to make sure we just weren't somehow selecting cases that developed antibodies, say, at an early age of life, and that really wasn't the case. Or diagnosed with diabetes either earlier or later in life. And again, they were well matched to the other cases that went on to develop diabetes. Again, HLA between the cases, subset of cases that we were able to identify early in life, again, very, very well matched. But we did find some interesting findings when we looked at that subset that we could identify roughly at 2 years of age. They had a predominance of specific islet antibodies. All of them had antibodies directed against glutamic acid decarboxylase. Nearly all of them had antibodies directed against islet-antigen 2, especially when compared to the rest of the cases. Not as much of a difference between those that developed insulin antibodies or those to zinc transporter 8. So I'll just give a brief summary of the talk here before we move forward, but T-cell receptor repertoires are very diverse early in life, become more clonal over time, likely due to infections and vaccinations. We were not able to find a distinct T-cell receptor V gene that distinguishes DAISY cases that went on to develop type 1 diabetes versus controls. However, we could identify a subset of cases early in life just with T-cell receptor beta chain sequencing from DNA that went on to develop antibodies in type 1 diabetes. Again, small numbers will need to be validated in the future, but quite interesting that T-cell receptor V gene usage was correlated to antibody profiles of GAD and IA2. So after going through all of that, and I'll say being a little shocked, disappointed that it wasn't as easy as we were hoping, that we could just find the a V gene predominance in cases that go on to develop type 1 diabetes, we decided to ask the question, can we identify known antigen-specific T-cell receptors in our data set? So maybe we just need to know more specifically what we need to look for. So again, this works very well in infectious disease, and just shown here are 2 articles from adaptive sequencing where they were able to sequence T-cell receptor beta chains from DNA in individuals with cytomegalovirus and make very good decisions about past exposure. And then obviously the pandemic went on, and T-cell receptor sequencing can readily identify those that have had SARS-CoV-2 infection and correlate T-cell receptors to neutralizing antibodies and severity of disease. So just for a control, we took all of our data and said, well, they're young children. They might not have had CMV at the age of 2 or 5. All these samples were collected pre-COVID-19. So we decided to look for influenza A specific T-cell receptors. These are predominantly CD8 and known to respond to pieces of hemagglutinin or matrix protein. So shown here are, again, cases, controls, number of individuals at a given time point, and then number of templates. This individual has more than several of the others. But we were very readily able to find influenza T-cell receptor sequences, over time, independent of disease. They're in cases and controls, and likely more as you get older in life, which makes sense because exposures do happen. So that was good for us to see. And I should mention too that we matched V, D, and J and CDR3. So this just wasn't CDR3. We wanted to match V, D, and J, especially as a junction region can make important contacts with peptide MHC contacts. So we then hypothesized that a subset of disease-relevant sequences obtained from the inflamed pancreatic islets of organ donors may be present in blood and accumulate through the stages of diabetes. So instead of influenza, we were trying to identify those receptors that might be important or localized to type 1 diabetes. So prior to this, we've been working with the lab of Maki Nakayama to study the residual T cells from pancreatic islets of organ donors that had developed diabetes and were newly diagnosed. This is through the NPOD program, Network for Pancreatic Organ Donors. And at the time, we had identified CD4 and CD8 cells that see pieces of insulin or preproinsulin. Just includes a signal peptide and C-peptide that eventually get cleaved before secretion of insulin. So we had 44 known alpha-beta T-cell receptors from the residual inflamed islets of type 1 diabetes organ donors that responded to pieces of proinsulin. And we just went back to our large data sets and said, could we find any of these? Were any of these shared or public? And so here are the 15 receptors that we found in at least one individual. Here are numbers of total templates, and just as you can see on top from controls, there were 11, obviously many more for this receptor that sees a piece of insulin B chain shown here. So we were quite pleased to see, 34% of the sequences were shared. And we went on to look at, well, when do we find these sequences? Are they early in life? Are they around antibodies? And so shown here are those CD4 T-cell receptor beta chain templates and CD8 on the right. Again, controls blue, cases red. As you can nicely see, these tend to accumulate after the development of antibodies and right before the diagnosis of clinical type 1 diabetes. CD8 maybe, but not as striking as CD4 beta chain templates. And then shown here, we were again trying to just localize this by stage of diabetes. So when a given case was antibody negative, lined up for their visit when they had seroconversion, so developed islet autoantibodies for the first time, and then that period of time where they're antibody positive, but before developing clinical type 1 diabetes. And again, some of these are present earlier in life, but the vast majority, again, not in all individuals, but a subset have accumulation of these pre-proinsulin reactive T-cell receptor after seroconversion prior to diabetes development. CD8, again, similar, more here before diabetes diagnosis. So we had sequenced individuals early in the disease course. We did want to try to validate some of these findings. And so harder to go and find a lot of individuals that have been followed over time, but we do have a large clinic that sees many children and young adults with newly diagnosed type 1 diabetes. And so we took some of those individuals, 143. So only sampled and sequenced once. Their median age was 12, very similar to when our DAISY cases developed diabetes, gender, ethnicity, diabetes for a very short duration, only 2 days. Did that mainly to limit any T-cell responses that may come from exogenous insulin, and the vast majority of these individuals had islet antibodies. It was nice to see that the sequencing was very similar between our DAISY controls and cases in terms of numbers, and we were able to find 8 of those 15 T-cell receptor beta chains in this separate cohort of new onset type 1 diabetes individuals. We found a lot of CD8 at the time of clinical type 1 diabetes onset. So that actually worked really well for us. We searched for 44 pre-proinsulin T-cell receptors, found roughly a third that were present in DAISY individuals. So we then wanted to do this at a much larger scale. We wanted to look for all islet antigen-specific alpha-beta TCR sequences, go back to our data sets and search for T-cell receptor beta chains. And so the team in my lab, we went through all of PubMed for the preceding 10 years, especially when T-cell receptor sequencing has really blossomed, developed, and made a lot of these studies easier. We wanted the T-cell receptor to be paired, and so have an alpha and a beta chain, and be antigen specific. And so we said, okay, you had the single cell clone and then sequence the receptor. You could tetramer multimer stain, single cell sort, and again, get the receptor. Some of these have just been crystallized, so trimolecular complex of receptor, seeing peptide and MHC. And then another way that this is done, and one that we use in our labs, are making T-cell receptor transductants. So take a given T-cell receptor of interest, put it into a TCR null cell line, and then test and determine peptide and HLA specificity. So we did that. We found a lot of islet antigen-specific T-cell receptors, also looked for viral receptors as well. And then we wanted to go back to our 3 cohorts of controls and cases longitudinally over time, and then our new onset cohort, so roughly a little over 350 samples. So here's what we were able to find going through the literature for alpha beta T cell receptor sequences that have a known islet-antigen specificity, CD4 on the left, CD8 on the right. So here are all TCRs, 243 that responded to some part of pre-pro-insulin, a lot to glutamic acid decarboxylase. HIP is a hybrid insulin peptide, a post-translationally modified peptide, strongly implicated in the pathogenesis of type 1 diabetes, and then some that were just 2 islet peptides. We found quite a few that were in at least one sample within our large data set. But then we said, well, we'd like it to at least be somewhat frequent, so greater than 1% of our samples, so at least 4 or more. And when we get down to those levels, you can see the numbers of all CD4 T cell receptor beta chains, 136, 18 for CD8, and again, insulin GAD and these hybrid insulin peptides were the predominant reactivities. Pre-pro-insulin and zinc were the predominant reactivities for CD8. And what I found quite nice was, 12% of these sequences are public, shared, and frequent. Again, I think most people know that the T cell receptor repertoire of a given individual, most are private, unique to us as a given individual. But really nice to see that we found a good chunk, 154 sequences, that could potentially be used as a biomarker of T cells that are public and frequent. So here is what the data looks like when we look over time for these different antigen specificities. So again, CD4 T cell receptor beta chains that see pre-pro insulin. Controls are in blue. Cases are kind of in this maroon color, and new onsets, again, a separate cohort off to the side. And we started to look by age of diagnosis. So again, both of these are present early in life. CD4 pre-pro-insulin reactive T cell receptor beta chains do seem to accumulate up to 9 to 12 years of age, and again are quite predominant in that new onset cohort, which will be a theme here. GAD reactive CD4 T cell receptor beta chain templates, again, quite a few early in life in both cases and controls. They tend to decline, and then there looks to be a pattern of maintenance in those that go on to develop type 1 diabetes. Again, large T cell receptor beta chain template numbers in our new onset cohort. And this was good to see. These pre-pro-insulin receptors do seem to accumulate up to a given point as the disease progresses, and again, are quite predominant in those newly diagnosed with type 1 diabetes. CD8 T cell receptor beta chain sequences, again, insulin on the left, zinc transporter 8 on the right. Again, we did not have nearly as many of these sequences to search for and that were public and shared. But again, somewhat of a decrease in controls or slight accumulation in cases. Again, strong presence in new onset. Zinc transporter 8, again, present in both cases and controls early in life, and a maintenance or expansion here later in childhood and early adolescence. So that was all receptors that we could identify, but we then wanted to know, can we identify those receptors that are really disease specific? So they're present more in those that develop type 1 diabetes or go on to develop diabetes versus controls. And so this was a bit challenging for us. So we had, again, very large data sets, well over 50 million T cell receptor sequences, and we knew frequency in our samples, and we also knew template numbers, and we thought both of these variables were quite important. And so what we decided to do was set up an XY plot subtract sample frequency in case minus control and template numbers in case minus control. And so I think shown here are the CD8, all of those islet CD8 T cells. So again, insulin and zinc transporter 8. And we were very much looking for those receptors that would be in the upper outer right quadrant to be more disease associated with type 1 diabetes cases or those in new onset. And so we highlighted the ones here that we thought were disease relevant in cases that progress to type 1 diabetes, so preclinical disease, and those at new onset type 1 diabetes. Then we did the same thing for CD4 T cell receptors, insulin on the top, again, cases, new onset cohort, and GAD shown on the bottom, again, cases that progress to type 1 diabetes and those with very new onset clinical type 1 diabetes. And again, just shown here were the receptors that, again, we thought were more frequent, higher template numbers. It wasn't always as simple as we wanted it to be. This receptor, obviously not overly frequent, but many, many, many more templates in cases than controls. And so we went through, made our selection of 73, what we'll call disease-associated T cell receptor beta chain sequences. And just shown here is multivariable plot. So those that see pre-proinsulin, GAD for CD4, CD8 for insulin, and CD8 for zinc transporter 8. The hybrid insulin peptides are included up here in the pre-proinsulin. Size relates to sample frequency, color relates to number of templates. Kind of a common theme is, again, these are very, very prevalent and predominant in new onset individuals. Some of these are present, such as receptor 1, again, very early in life. They stay present, but may decrease in template number. Some of these are quite interesting, again, present in kind of lower amounts early on, but again, that expansion as the disease progresses and antibodies have developed. So this is all very nice. You can say, good, you found these receptors and even these 73 that you like, but we did want to try to see if these were at all starting to be clinically meaningful. So we have 73 disease-associated, potentially disease-relevant T cell receptors. Do they mean anything to the disease course or progress? And so shown here are the age at type 1 diabetes diagnosis, clinical onset. And these are the number of those unique 73 sequences. So this individual here has 16 of those 73 and developed type 1 diabetes somewhere at around 7 years of age. And there started to be a trend towards the more numbers of these disease-relevant sequences that you had to age at type 1 diabetes onset. Template numbers didn't quite work as well. It was, again, more the numbers of these disease-relevant T cell receptor sequences correlating to a younger age at diagnosis, which was nice to see, and I think a really good start to trying to use T cell receptor sequencing as a marker in type 1 diabetes. So just a few take-home messages. It's feasible to sequence the T cell receptor beta chain repertoire from small amounts of peripheral blood very early in life and in childhood. Forever, we have wanted to track and monitor T cells in diabetes, and it's very challenging. Obviously, functional assays with tetramer staining, proliferation, ELISpot are great, but there are challenges in terms of blood volumes, especially when studying really young subjects. A subset of islet-antigen-specific T cell receptor beta chains are public and can be tracked during the development of type 1 diabetes. Those disease-relevant receptors, those found more frequently in diabetes than HLA age-sex matched controls and blood do start to correlate to age at clinical diagnosis. And again, my opinion, but I think T cell receptor sequencing can provide a powerful biomarker to really monitor progression towards clinical type 1 diabetes. And especially since we now have immunotherapies, at least one approved to delay onset of diabetes, many more being developed and in clinical trials. I think having a marker of active disease or increased likelihood of progression is much needed. So some of our next steps and future directions of what we're thinking about is how can we further improve T cell receptor beta chain sequencing to predict at an individual level when someone will develop either islet antibodies or clinical type 1 diabetes. Approach we're using is to just simply target the specific V genes used by those islet antigens. The entire repertoire obviously is not used to target things like insulin, GAD, zinc transporter 8. And so focused sequencing, deeper sequencing may provide benefits. It'd be very interesting to use machine learning techniques, I'll say on our large data set and many others that we've worked with part of the AIR community and Felix to really develop. We now have a T1D T cell receptor repository on the iReceptor gateway. So all of this data, clinical metadata there. I should say also Todd Briscoe from the University of Florida has put a tremendous amount of data in as well. And we are really working hard to build this up and not just have sequencing from peripheral blood, but sequencing from tissues, spleen, pancreatic lymph nodes, and even islets, and having all of this data and important clinical metadata available to the community. T cell phenotype is very important, not just in type 1 diabetes, but many disease states. And there are ways to infer, especially some phenotypic changes from just the T cell receptor sequence. There are hydrophobic amino acids in CDR3 that really promote targeting self and can have a regulatory type of phenotype, also amino acids in CDR1. And so some of our next steps are to build that scoring-based system in to try and glean some T cell phenotype from T cell receptor sequencing. And then it's not lost on us either. We've very much been doing this in disease progression and natural history, but to measure T cell receptors, especially those that are disease associated pre and post immunotherapies from a lot of our trials. So with that, I'll stop and happy to take questions.
Thank you very much, Aaron, for this very nice and credible work on children and type 1 diabetes. I see that there is already many questions on the chat. So maybe Gore, you can directly ask your question to Aaron, your questions, because you have several.
Okay. Very nice talk, very nice data. Looking forward to look at it also. My questions were, for the first part were, as I listed, I mean, first of all, I didn't understand if you sequenced separately CD8 and CD4. From the second part, I understand you did, but I didn't.
So we did not subset cells first. So these were just cryopreserved PBMCs, Buffy coats, where we extracted DNA. And so we did not, we sequenced the full T cell receptor beta chain. Now, when we showed CD4 and CD8, I think that was from a known receptor. So alpha beta CD4 T cell that sees a piece of insulin presented by, say, HLA-DQ8. And we went back and matched that receptor. But we did not subset cells and sort from those subsets.
Okay, so the first part is just bulk, like everything together, PBMC, right?
Got it, yes.
Yeah. Because I mean, there are substantial differences in the V gene usage of CD4 and CD8, you know, and when you put them together, it's very hard to see changes between different cohorts. That's what I asked you. The second question there was regarding other features that could be used for classifications, not only V genes. Like I suggested biochemical properties of CDR3, but there are other like motif finding algorithms that can differentiate between two repertoires. Did you try to look at these kind of features?
So we have not done that in depth as much. I'll say, you know, our strength as a lab and as a group is really identifying T cell receptors, antigen specificity, you know, the machine learning and using some of those other algorithm-based programs is not not as much as our strength. And so I did, you know, kind of put this and want to put it out there that it should be done. It needs to be done, and happy to help and collaborate in any way I can to see that happen. And then also knowing too, it's great that we were able to do the work, but with more data comes a lot more power, especially good data where you know where it comes from in terms of individual stage of diabetes, antibody profiles. I think real inroads can.
Definitely. I mean, if this data is already available, I mean, my group will be happy to look at it, and I'm sure others as well.
Yeah, so I guess it is available. It's in the adaptive immunoseq, but really which is again fine and happy to share with anyone, but we wanted to be able to combine it with other large data sets in the community, and hence why the iReceptor gateway. Trying to
so speaking of the adaptive platform that you use, I mean, you showed the differences in V gene usage, but since adaptive is sequencing only a very partial region of the V gene, many of the alleles cannot be differentiated.
You're correct. Yes. So you're right. There's especially like TCRβ6. You don't get down to you can do gene, but it's harder to do the allele level.
Yeah, which links to actually what Felix replied to my comment, whether you think or you have evidence to think that there are some differences between the polymorphism that in the individual carries, like, you know, a higher tendency to have T1D if you have allele of some allele or not.
So that's a great question, something we have thought about, especially when we wanted to go. I mean, ideally, we'd love to sequence, you know, early in life, DNA, think whenever, and be able to say diabetes, yes, no, or even antibodies. And so I think some of what we started with and tried to do lays the foundation to say, boy, there, at least in TCRβ4, there seem to be some differences. So now can we go back, you know, deep sequence, say design primers, maybe not like adaptive, and look at the whole sequence. So I very much agree with that.
Okay, so last question before I need to let also other people ask. Regarding the lab which showed some significance numbers. If you can go back to this.
Sure. Yeah.
Yeah. So here, I mean, essentially, you're looking at the number of unique TCRs as a function of age of diagnosis, and you see some kind of trend. But did you try to use this for like a classification task? You know, because here you just see the cases, right?
Yes.
But can you use this data to actually stratify between cases and controls? Did you try to do this kind of, you know, machine learning task? Like,
so that's kind of where these. So we kept kind of funneling down, and so I'll say these receptors are very much more frequent and higher template numbers in cases compared to controls. And then obviously there are some too that are kind of present throughout the whole disease course. So say early on when you have antibodies, and then even in that new onset cohort. So it's not a complete overlap. I understand very well that there is a it's 049 trend toward significance, obviously, but gives me confidence to say, all right, we're on the right track. So how do we improve this? You know, how do we make it better? And I was trying to do a good amount of that with the kind of next steps. You're asking a lot of really good questions. Too, in terms of polymorphisms, phenotype, I really do want to go after. I've looked a lot at that. I kind of would really like to know what, say, conventional memory T cells look like in young children, and regulatory in terms of receptor usage, because everything I look at, I'm like, well, that's nice. You've had cancer and you're, quite a bit older. And so I do see somewhat of a need to say, what does the T cell receptor repertoire look like in antigen experienced, naive, regulatory cells from children with a genetic background for diabetes.
Okay, thank you very much. So it's really
Thanks for the great questions.
So Alex has also some questions.
Hi. I had a question that was related to Gear's point, which was just talking about acidic amino acids in the CDR3 beta, but I really wanted to ask more about in your controls. I was wondering if you have any of your patient population having the predominant protective DR2 haplotype in conjunction with DR3, DR4, and it'd be really interesting to see if the TCR beta chain sequences of those protected people have their own particular repertoire usage that you could then correlate and say these TCRs are perhaps responsible for dominant protection, something like that.
So that is a great question, very insightful. So unfortunately here, all these individuals have, predominant genetic risk, DR3, DR4, DQ2, DQ8. I don't believe any of them have DQ6 or 602. But in other work of the lab, we're very much interested in DQ6. It provides dominant protection for type 1 diabetes development. So the odds ratio is only 0.03, whereas if you have DQ8, it's roughly around 10 or 11. And so we are very interested in, again, peptides that may be presented, T cell responses in the repertoire, especially in those individuals that have really dominant protection from an autoimmune disease.
And then, Aaron, I just had one more question. So I think Mia is also sequencing BCR repertoires. Is this part of the DAISY study?
So good question. So we just received a grant from the Helmsley Charitable Trust to do just that. So we do want BCR sequencing from these same individuals, to have both TCR BCR sequences. I again would like to hypothesize and hope there's increased power with at least heavy chain sequencing.
So one really quick point on that is that in infectious disease, there's all kinds of antibodies that show up that are extremely present. They're among the most abundant antibodies that show up in the infectious disease, but they don't bind to the target. And some of those antibodies seem to be anti-idiotype antibodies, where if you look at the CDR3, they actually hold a motif that looks very similar to the presented peptide that the TCR is eliciting. So it's this bizarre forward feedback loop that's happening in the germinal center. But you can mine those CDR3s and perhaps pull out antibody sequences that then correlate to TCRs. And you already know that there's these autoantibodies that are showing up. But perhaps you could catch these antibodies that would otherwise not bind to a useful target prior to detectable serial conversion, and then be able to use TCR sequencing to kind of look for that. And you already know many of the TCR epitopes in type 1 diabetes, so you would kind of know exactly what to look for in the BCR sequences.
That is a great idea.
So I have a question following up on this late point. So in the first part of your presentation, you showed this subset of cases that had some V-beta, specific V-beta usage. And you showed also that there was kind of an association of those subsets of cases with the GAD antibody seroconversion. So I'm not sure. Maybe I did not get the point, but did you check in this subset of cases this increase of TCR unique specific TCRs for the different pancreatic islet antibodies, and is there a difference with the other cases?
That's a really good question. We actually have not done that. We do need to go back and do that, especially think T cell receptors in those 11 individuals. We haven't gone back. We haven't gone back and have done that. We really should, because it might help.
It might help and see eventually maybe the correlation between BCR, TCRs, antibodies, and specificities.
Hopefully in another, give us some time to get the BCR sequencing, but hopefully we'll do that and come back and share that with you in the future, or me.
Okay. Well, I don't know if there is other question in the audience. Gur has a last comment.
Although I think Felix raised his hand. But I just said that just regarding I mean, you mentioned that it's hard to take blood from children, which I understand. And actually, maybe, you know, it's more expensive, but if you go to single cell sequencing, you might get more insights from, you know, these small quantities of blood.
Yeah, we have thought about that. You know, the unfortunate frequency of some of these islet reactive T cell receptor sequences are, you know, 1 in 50,000 to 1 in 100,000, and it gets hard to capture enough of those cells without some sort of enrichment ahead of time. Right now, again, everyone is paying attention. We'd love to single cell, you know, alpha beta sequence and RNA-seq on, you know, obviously not tens of thousands, but hundreds of thousands of cells.
Yeah, I agree with you. Okay, so no other additional comments or questions? If not, thanks a lot, Aaron, for your presentation, and thank everyone for the questions. So now we will move to Julien's presentation. All right.
Can you see my screen?
Yeah.
Yeah. All right.